Okay, morning everybody. So let's get started. Um, Today we're going to finish the lipoprotein metabolism and regulation of a cholesterol homeostasis. Um, and I think the Thursday we're going to talk about fatty acids, essential fatty acids. That should be the last section, right, of the lipid metabolism. And then next Tuesday we have a review session, similar like we did for carbohydrate metabolism. I go through the confusing slides for like an hour or so, and then just leave some time for you guys to have questions. And this Thursday, I think maybe we will have some extra time. I think we probably will finish earlier. I can open for question if you have, so you can get prepared your question. And also, it's up to you guys. And also, I think you guys probably need some time for working on your review papers or something like that. The review due will be next Tuesday, right? No, sorry, this Thursday, this coming Thursday, the peer review paper, right? Okay, so fatty liver, I think we stopped here, right, last class. So fatty liver, in terms of the cause, is kind of imbalance between triglyceride formation and triglyceride export. So the main reason, the cause of a fatty liver disease is the accumulation of triglyceride in the liver. So we either due to the increased triglyceride synthesis in the liver, or it could be due to the decreased triglyceride export, right, from the liver. So this is a cause, the imbalance. And we also talk about the type of fatty liver disease. That actually two major types. One is called non-alcoholic and the other alcoholic. So non-alcoholic, the reason or the causes of non-alcoholic liver disease can be very complicated, but alcoholic fatty liver disease is relatively simple. It's just caused by the alcoholism. So the first type of fatty liver disease, non-alcoholic fatty liver, we can caused by two major reasons, right? Also, we can thinking of the two main type of non-alcoholic disease. So the first cause for the fatty liver disease is the increased triglyceride synthesis, right? Remember the imbalance. So increased triglyceride synthesis can be due to the increased level of a free fatty acids in the plasma, in the circulation. If we have too much or too high levels of fatty acids in the circulation, then liver will pick up fatty acids and synthesize triglyceride, right, in the liver. So this is the main reason that causing the increased triglyceride synthesis. And high levels of plasma fatty acids can be caused by, several reasons, several disease conditions. For example, diabetes is one of the major problems that causing fatty liver disease, causing the high levels of fatty acids in the plasma and fatty liver disease. I think we already talked about it, right? The reason, remember that the diabetes can also cause the ketogenesis, right? Ketoacidosis, because of the utilization of glucose decrease in the diabetic condition. So that will promote the fat mobilization, so allows to promote a lot of triglyceride and dehydrolyze and release fatty acids into the circulation, so raise the blood levels of fatty acids. So in the diabetic conditions, high fatty acids level in the circulation is high. So, and this is a cause that the liver can pick up fatty acids and synthesize triglyceride and store in the liver. And it also can be due to the chronic intake of high-fat diet, right? So if we consume a high-fat diet and for long term, then we always have a high levels of fatty acids in the circulation, so that this will also causing the fatty liver disease. And the second major cause for the fatty liver disease is the decreased triglyceride export. We know that liver is not the primary storage of triglyceride, right? Adipose tissue is. So triglyceride synthesized in the liver needs to be exported and transported to the adipose tissue for storage, or transported to the skeletal muscle for energy under the fasting condition. So for some reason that triglyceride synthesized in the liver cannot be exported, then we're also causing the triglyceride accumulate in the liver. And for this cause, there are actually several conditions that can cause the decreased triglyceride export. So overall, the major reason is the metabolic block of the production of plasma lipoprotein can be the major reason for the decreased triglyceride export. Remember, triglyceride transporter needs lipoproteins, right? Particularly the VLDL. So if for some reason, pathological reason that lipoprotein VLDL cannot be synthesized, then the triglyceride cannot be exported, right? Cannot be transported out of the liver. So here I listed like 4 reasons for the decrease or for the problems with the lipoprotein synthesis. First of all is the apoproteins. We know that lipoprotein, for the full function of a lipoprotein, also needs a apoprotein, right, on the surface of a lipoprotein. So for some reason that apoproteins cannot be synthesized, which is pretty common in some kind of a genetic disease, then lipoprotein cannot be synthesized as functional, right? And the second is the synthesis of lipoprotein from the lipids or apoprotein, not just the apoprotein, but some other reason that the triglyceride cannot be packed into the lipoprotein, that can also cause a problem. And third is the lack of phospholipids provision. We know that for synthesis of a lipoprotein also needs the phospholipids, right, for making the monolayer surface of a lipoprotein. So if some reason that phospholipids cannot be made or cannot be provided for the lipoprotein synthesis, then it also could be the the problem. And lastly, the impaired secretion of lipoproteins. So even you are able to synthesize lipoprotein, but lipoprotein cannot be secreted, right? So it could be all causing the problem that triglyceride cannot be exported from the liver, right? So these are the 2 main causes for the development of fatty liver disease, or in other words, for the triglyceride accumulation in the liver. So liver synthesis, right? I mean, you have a more triglyceride synthesized, or you have a decreased triglyceride export. All right, so the second type of fatty liver disease, which is alcoholic fatty liver. Um, so pretty simple. It's just because of consume too much alcohol. So alcoholism, always related to the fatty liver disease. As reason for alcoholic fatty liver disease, it could be the impaired fatty acid oxidation and increased lipogenesis or triglyceride synthesis. Here is the how alcohol ethanol is metabolized. So ethanol in the body can be metabolized by the enzyme called alcohol dehydrogenase, and this reaction produces a NADH. So alcohol is metabolized and produces NADH. So we know from the metabolism of a fatty acids, carbohydrate, and protein can also produce NADH, right? So NADH is very important reducing equipment and go to the mitochondria, do the oxidative phosphorylation, and generate ATPs, right? So if alcohol is metabolized and produces too much NADH, that this NADH molecules can compete with the NADH that generated, the normally produced from the nutrient metabolism. So this will increase NADH levels generated from alcohol metabolism can inhibit the fatty acid oxidation, right? So you can see this sort of the pathway and how alcoholism can increase the triglyceride synthesis and fatty liver disease. So in general, the alcohol is metabolized and produce NADH, which compete with the NADH from normal nutrient metabolism, and then inhibits the fatty acid oxidation. Of course, there's lots of other things happening, but we don't need to know. But just the increased NADH can, stimulate or can causing some signaling pathway or molecules generate and then inhibits fatty acid oxidation. So fatty acid oxidation is inhibited, and the more fatty acids will be used for synthesis of triglyceride. That this is causing the triglyceride accumulation in the liver, right? Okay, so now let's talk about the cholesterol homeostasis. Remember we talk about the regulation of the lipid metabolism. For the triglyceride metabolism, we already mentioned that the dietary lipids play a pretty important role in regulating circulating triglyceride metabolism, right? And we also mentioned that the ApoC2, remember the apoprotein, which is an important co-activator of the lipoprotein lipase, LPL. So we give kind of the example, right? So the genetic problems in the ApoC2. So if patients with ApoC2 deficient, then the LPL cannot function normally. So then cannot hydrolyze triglyceride in the VLDL and the chylomicron, and will causing the increase the triglyceride level in the circulation, and also increase chylomicron and VLDL levels, right? Remember this. And now we switch to the cholesterol metabolism and homeostasis. So cholesterol mainly levels in the cells and also in the circulation, mainly is regulated by the two important lipoproteins, remember, right? LDL and HDL. So LDL is more related to the circulating levels of LDL. And HDL is more important, regulates the the homeostasis between the plasma and the cell, peripheral tissue cell levels of cholesterol. So let's talk about the plasma LDL or cholesterol homeostasis first, right? So how cholesterol levels in the circulation be maintained at a normal range. So how this can be regulated. So first of all, we know the cholesterol present in the circulation mainly as the LDL, right? As the LDL cholesterol. So increased plasma cholesterol is mainly associated with a decreased uptake by the cells, right? So try to remember this mechanism. So the uptake of LDL from the circulation into the cell is the main mechanism, right? As the most important mechanism that control the levels of LDL in the circulation. And we also mentioned the LDL receptor, remember, right? LDL receptor can be present in the liver and also peripheral tissue. So, but majority of LDL in the circulation is picked up by the LDL receptor in the liver, so by the liver, and some percentage of the relative small percentage of LDL they pick up by the peripheral tissue, right, through the LDL receptor mechanism. So that is in the normal condition. So the so in some disease conditions, for example, the decreased LDL receptor for some reason in the liver or some peripheral tissue, that the LDL cannot be picked up by the liver, right? So that will causing the increased LDL cholesterol level in the circulation. So increase the blood cholesterol levels. And the other conditions, disease condition or pathologic condition is that the defective, the ApoE or ApoB100. Remember, what are the function of the ApoE and ApoB100 in terms of regulation of the LDL uptake? Say that again. Yes, the receptor binding, right? The ApoE on the surface of LDL or ApoB100 on the surface of LDL and HDL are very important. The apoprotein that can be recognized by LDL receptor and then be able to pick up by the liver or the peripheral tissue cell that has the LDL receptor, right? So if we don't have enough ApoE or ApoB100 on the surface of LDL or HDL, or IDL actually, HDL remnant, right? IDL surface, then the LDL cannot be picked up, right, efficiently by the liver or peripheral tissue. So that will also causing the increase LDL. So you have a lot of LDL cannot be picked up by the tissue and then remain in the circulation. So try to remember these two main reasons or problems that can causing increase the plasma levels of LDL or LDL cholesterol. One is the LDL receptor deficient, a decrease, and the other reason is the ApoB100, ApoE protein defect can also cause the similar issues, right, raising the LDL receptor in the circulation. So again, this are the main regulate mechanism that control the plasma levels of LDL cholesterol. So just wanted to connect this to the real life issues. So I think I mentioned this, or you guys also had question about whether, dietary cholesterol affect the cholesterol levels. So I think that the genetic factors could be the most important, right? So if the normal people are thinking we should be able to handle with the cholesterol if we, consume too much. But the problem is, the patient, if they don't have enough, this regulating mechanism, then it should be easily causing the problems, right? Okay, now the second aspect of the cholesterol homeostasis is in the cells. The LDL cholesterol are not only present in the plasma but also in the cells. So how in the cellular cholesterol levels can be maintained at the normal range. This is also important and directly related to the atherosclerosis, right? So this little bit complicated as compared to the plasma LDL homeostasis. But let's look at this. So this represents a cell, right? Particularly here, particularly in the liver. And the inside of the cells, there is the free cholesterol pool, right? And also cholesterol esters. So free cholesterol can be re-acidified to cholesterol esters, or vice versa. Cholesterol ester can be, break down back to the free cholesterol and fatty acids, right? So the free cholesterol pool also need to be balanced. We don't want it to have too much cholesterol inside of cells that will cause problems. So and there are actually two different sets of pathways that contribute to the free cholesterol pool. First is the I think I labeled it differently, right? The label like black circle and white numbers, and this represents the pathway that adding free cholesterol to the pool, and the box square and the red numbers, and this represents the pathway that remove the free cholesterol from the pool, right? So let's focusing on the pathway that can add the free cholesterol to the pool. So the first pathway is the the uptake, right? If the cell uptake a lot of LDL from the circulation, then you contribute the free cholesterol to the pool. So you're adding a lot of cholesterol, free cholesterol to the free cholesterol pool, right? So for example, if we uptake a lot of, the uptake is increased, then you have increased the free cholesterol pool. And the second pathway is the synthesis. Cholesterol cell, we know that almost any type of cell is able to synthesize cholesterol, right? So if cholesterol synthesis pathway is somehow upregulates, and then it also can add the more free cholesterol to the cholesterol pool. And the third pathway is the cholesterol ester. For some reason, being break down to the free cholesterol, and this is the third pathway that can also add free cholesterol to the cholesterol pool, right? So three pathways, if they are activated, can causing the increased free cholesterol inside of cells, right? Now let's look at the other three pathways that can remove free cholesterol out of the pool, which is good to keep balance, right? So the first pathway is the ABCA1 mediate the efflux or the output, right, or the export of a free cholesterol. And this is a pathway we talk about. It's very important, the HDL cycle, remember the cycle, or the reverse cholesterol transport pathway. So remember the pre-beta HDL is the HDL that can pick up the free cholesterol from the peripheral tissues, right? And this is the pathway, you know, the ABC1 is the transporters, right? Transporter free cholesterol from cells to the beta HDL, and then become alpha HDL and transport cholesterol to the liver. Yes, question? Yes, the same. I don't know why they miss, you know, ABC. The same, yeah, ABCA1, right? ABCA1 protein. Okay, and the second pathway is the, you know, free cholesterol can be utilized for the synthesis of the sterols, right? Particularly in the liver or for the bile acid synthesis. So this is the second pathway that can remove the actual free cholesterol from the cell. And then the third pathway can be reacidified to the cholesterol esters, right? It can also reduce the free cholesterol. So this different sets of the pathway need to be balanced, and then can keep the free cholesterol at the normal range inside the cell, right? Yeah. Yes, yes. So remember we have another one, and the same enzyme that present in the plasma, right? So try to remember. Yeah, this one's ACAT, right? It's inside of the cell. Yeah. Yeah, LCAT, right. Thank you. All right. So, so now let's talk about under the disease conditions, right, the pathologic conditions. So if something happened that, you know, for example, 5 reasons that can cause increase the intracellular cholesterol levels, which is not good, right? And for example, first of all, it's uptake. If for some reason we uptake too much LDL into the cells, that also causing increased free cholesterol in the cells, right? So increased uptake of LDL is the first reason. And the second, for some reason that we synthesize too much cholesterol inside the cells, that can also contribute to increased free cholesterol. And hydrolysis of the cholesterol ester too much, that will also contribute to increased free cholesterol, right? And for some reason that we cannot export free cholesterol from the cells, for example, ABCA1 somehow deficient, right? So then you cannot transport free cholesterol from the cell and to the beta HDL, and cannot remove the free cholesterol, right? So efflux of the free cholesterol decrease, that will also cause an increased free cholesterol, or utilization of the free cholesterol. For example, you know, free cholesterol cannot be utilized for synthesis of sterol or bile acid. So bile acid synthesis pathway somehow is blocked, and also will increase free cholesterol inside of cells, right? So try to remember the 5 reasons that can cause increased free cholesterol inside of cells, and which can be related to the development of atherosclerosis, right? Okay, yeah, so this just kind of, you know, the reason, for example, LDL receptors or cholesterol synthesis pathways and ACAT activities, right? Changes can also change, you know, this synthesis or uptake or the reacidification process, right? Any questions about this intracellular cholesterol homeostasis? Is that clear? Right. So once you clear about this, two different sets of pathway that regulates the cholesterol pool, then you can just easily to understand and to make clear. I think it should be easier to understand, right? Okay, so then cholesterol kind of clinical relevance, the cholesterol homeostasis. First is cholesterol we know is closely associated with the coronary heart disease, right? So if you can look at this diagram, you see the y-axis is the heart disease mortality, and x-axis represents the serum cholesterol levels. So between 35 and 5 millimole per liters is the normal range of the serum cholesterol, right? And you can see they have a lower rate of the cardiovascular disease mortality. And if the serum cholesterol level increased from 5 to 6.5 millimole per liters, and this range is considered kind of borderline, but you already see the mortality of the cardiovascular disease is increased, right? And if the serum cholesterol level is above 6.5, then you see the significant increase in cardiovascular disease mortality. So that is, everybody probably understands this, right? The cholesterol levels definitely have a positive relationship with the cardiovascular disease development. So we always wanted to keep our cholesterol levels low. And now the saturated fatty acids, the relationship between saturated fatty acids and the serum cholesterol. So if you can also see the positive relationship that if we consume more saturated fat, and we can increase the serum cholesterol levels, right? But polyunsaturated fatty acids have a negative, correlation with the serum cholesterol. So if we consume more polyunsaturated fatty acids, we can reduce the cholesterol levels. And this all the study from the the epidemiological studies. And dietary cholesterol is also positive associated with the serum cholesterol level, right? We consume too much cholesterol, definitely we will increase the cholesterol levels. Okay. Now let's talk about atherosclerosis, right? So atherosclerosis is the major risk factors for the development of atherosclerosis is elevated plasma cholesterol levels, right, and not the triglyceride level. So triglyceride level is independent factor, meaning that it's the elevate the increased cholesterol level in the circulation is the major risk factor for the development of atherosclerosis, right? And the problem of atherosclerosis is due to the deposition of the cholesterol or cholesterol esters, and that will promote the formation of atheroma. So atheroma formation is a major problem of the atherosclerosis, right? And atheroma basically is formed from the cholesterol and cholesterol esters, right? And this causes the blocks of the blood flow and leading to the heart attack, stroke, and some other peripheral vascular disease. So aceroma or atherosclerosis is really the major problems of related cardiovascular disease. And it's just, the slide just show you, the aceroma as formed in the artery wall and can block the blood flow and affect the, the blood supply of the heart, particularly that can cause a lot of problems, a heart attack or, or vascular problems. And in terms of the aceroma formation, there are several reasons. First is the endothelium damage together with the inflammation. And that endothelium damage probably is associated with some high blood pressure. So if you have a chronic high blood pressure and causing the endothelium damage of the artery walls. And this damage can trigger the infiltration of inflammatory cells into the damaged artery wall. For example, macrophages and monocytes, right, the immune cells. So a lot of immune cells will go to the damaged site of the artery walls. And then the lipoproteins, particularly LPL, right, is is a major lipoprotein that can retain in the local area of the damaged artery wall, right? And this, the LDL that are checked by the damaged site is caused by high levels of plasma LDL, right? And this LDL at the local site can be modified and oxidized, right? So oxidized or modified LDL then can be recognized or can be uptake by the immune cells, particularly macrophages, right? And the uptake of a lot of LDL cholesterol into the macrophages, and this macrophages then become the firm cells. So you have a lot of firm cells that filled with the large number, the large amount of LDL cholesterol, then can block, form the aceroma and block the blood flow, right? So actually, there are the major two reasons, right? First is the damage of endothelium layer of the artery wall, and the three major reasons, right? First, the damage of the endothelium layer of the artery walls, and second is the inflammation, right? So you attract a lot of immune cells, macrophage and monocytes, and lastly is the increased LDL levels in the circulation. So that will allow macrophage to pick up a lot of LDL, right? So three reasons that okay, so cholesterol, I just wanted to go back this slide. Remember that the peripheral cells, and here we're talking about macrophages, right, can pick up LDL, and the normal pathway that for LDL pickup is through the LDL receptors, right? Remember, so LDL receptor that requires ApoB100, ApoE proteins that to recognize receptor and pick up by the receptor-mediated mechanism, right? LDL. This is a normal LDL pickup pathways. But under the disease conditions, like I just mentioned here, during the development of atherosclerosis when we have high levels of LDL, um, and this LDL can be, when it's reached the local site of damage, local damage site, then LDL can be oxidized, right, to oxidize or modify to oxidize LDL. And this oxidized LDL can be picked up by the non-regulatory pathways, or also called the scavenger receptor. Remember we mentioned scavenger receptor, right? So in the macrophages, the macrophages also have a scavenger receptors, and that scavenger receptors can recognize oxidized LDL and pick up the oxidized LDL. So this can be independent of the normal LDL receptor pathways, right? All right. Can we take a 10 minutes break? Okay, so I know you guys quite busy, right? And to for the paper due this Thursday, some of you mentioned that asking if we can postpone the due date till Friday. Would that be happy? Okay. All right. So, so the due date for peer review, right? Peer review papers due on this Friday, right? I'm going to change the the settings. So, um, and what else? Oh, so and I think this particularly the lipid metabolism in this class is very complicated, even more difficult than the carbohydrate metabolism. So just feel free, if you guys have any questions, and just come. My door is always open. You can just come in and ask questions, and or you want me to clarify something. And also, I mean, if you have a specific time you're available and you wanted to make sure that I'm available, and you can email me to make a appointment. Otherwise, I mean, just open door policy. You can come ask any question. And also in the class, I mean, you guys feel free to ask questions. I know it's very complicated. So, and other thing is, I think for the how to study the metabolism, I think the approach probably is most important. Try to understand, right? The big pictures, and then you go to the details and understand the details. And I think we mentioned that, not like, remember this name of the enzyme or the reaction, and try to remember the whole pathway, and we just specify some key enzymes or, key reactions, and try to remember these reactions. And most importantly, the regulation, right? How, this pathway is regulated. So once you understand how, this pathway is regulated by, for example, by insulin or by other complementary regulatory hormones, and they should be very easy to understand this key enzyme in that particular pathway should be, either up or down regulated. And and then when you understand the details, and then you come back to the big pictures, right? So I always ask you to study, thinking first of all, the top level should be the two different physiological conditions, fat or fasting condition, right? And then the second level be the hormones. What type of hormones that increase or decrease in the fat or fasting conditions? And then the third level is which metabolic pathways is increase or decrease. This approach should apply to all the three nutrient pathways, right? The carbohydrate, lipids should be same thing. So and then go to even more detailed, which specific enzymes that regulate this pathway, for example, lipogenesis or hydrolysis or lipolysis, right? And then you connect, and connection is also very important, right? See the the enzyme, for example, in the fat conditions, we know the insulin is up, right, regulated, and then for the lipogenesis, it should be increased. And then you also need to understand what about the pathway in the that being upregulated in the fasting condition. They are kind of interrelated, In the fat condition, you should activate the pathway in the lipogenesis, right? And you should inhibit the lipolysis. In fasting, lipolysis should be increased. So just try to understand things from the different levels, and go to the details. And then you also very important to connect this two different pathway in two different physiological conditions. One up and the other should be down, right? Something like this. So understanding is most important, that will help you to memorize the stuff. And also sometimes if you don't really remember this, but if you just do the approach like I said, it should be also, you can just make a prediction or make the guess. Sometimes it will work, right, in the for the exam, stuff like this. Okay, so atherosclerosis, right? I want to try to finish this. So as I mentioned, and so this diagram give us a better picture of how atherosclerosis is developed. And I mentioned the three major things. First of all, you have to have a higher LDL levels, right? So if normal health person, we should not be able to, you should not develop atherosclerosis, right? Because we have a normal levels of LDL. So only under the certain physiological conditions, when the somehow LDL levels goes high, and then also damage, right? Artery wall damage, and this usually associated with the chronic high blood pressure, right? Your person with the high blood pressure, that's very easy to cause for the long term, right? It can cause the artery wall damage, and then the inflammation, right? So three things come together, then can push the development of atherosclerosis. So here, if you look at this diagram, so this represents the endothelial cell layer of the artery wall, right? And first of all, you should have some damage, right? The endothelial wall, and then causing the accumulation or attract the infiltration of the immune cells like monocyte and macrophages, right, into the damaged site. And the important thing is the increased LDL level in the circulation. So this LDL and in the at the damaged site can be oxidized, right, to oxidized LDL, and this oxidized LDL can be picked up by the macrophages, right? And so through, again, I mentioned through the scavenging receptor mediated mechanism. So then you have a lot of cholesterol being taken up by the macrophages and then accumulate inside of macrophages. And this macrophage filled with the cholesterol, we give the name called firm cells, right? So you have a lot of firm cells and build it up, and then causing the formation of the atheroma. So again, just try to remember these three things: high levels of LDL, damage of artery wall, and inflammation or macrophage infiltration into the cells, and LDL oxidized and uptake by scavenging receptor mediated mechanism into the macrophages and firm cell formation, right? This whole process, and for the atherosclerosis. Any questions about the atherosclerosis? No? Okay, now we have three case studies that, as I mentioned, that the genetic factors are the most important factor that can regulate, the cholesterol metabolism and can relate it to the disease development, right? So let's look at case one. Case one is called tangent disease. Tangent disease, the reason is the mutation of ABCA1 genes. Remember ABCA1? Where is the ABCA1 located? Peripheral cell, right? Cell surface. And what is this protein does? Yeah, efflux of cholesterol, right? Transport free cholesterol from the peripheral cells and loaded free cholesterol to pre-beta HDL, right? Remember this? So ABCA1 gene deficient is the cause of the Tanginger disease. And what happened to the patient with Tanginger disease in terms of atherosclerosis? Do you think this patient will have a high risk to develop the atherosclerosis? So first question is, what about the alpha HDL in the circulation in the patient? What do you think of alpha HDL levels in the circulation of the Tanginger disease patient? Low? Yes. Low, right? Because the alpha HDL is converted from the pre-beta HDL, right? By loading free cholesterol from the peripheral cells. So if ABCA1 deficient, then free cholesterol cannot be loaded until the pre-HDL. So alpha HDL cannot be formed, right? So this patient have a lower alpha HDL levels or even absent in the plasma. And what about beta HDL? High, right? So beta HDL cannot be converted to the alpha HDL, so remain high levels in the circulation. And risk for the atherosclerosis, high, right? Because you cannot transport free cholesterol out of the peripheral cells, particularly in the macrophages. So you have a lot of cholesterol accumulated in the macrophages and forming the firm cells and causing the atherosclerosis, right? So a question here.
How is this diagnosed or spotted? People just have really high levels of lipids in their plasma?
Yeah. So they have very high levels of, the have actually lower HDL, right? Alpha HDL and higher beta HDL. So you can measure the profile of lipoprotein, HDL, and so you can. All right. So case 2, familial lipoproteinemia. So in this type of patient that they have a defect LDL receptors or mutation in ligand region of ApoB100. So what happened to the patient in terms of atherosclerosis? First, the LDL levels in the circulation. What do you think the patient, if they don't have enough or have a lower LDL receptors, or they have a mutation in ApoB100? What do you think of the LDL levels in the circulation in this type of patients? High, right? So because circulating LDL cannot be picked up by the liver or peripheral cells because of the lack of LDL receptors or ApoB100. And what about VLDL remnant levels in the patient? VLDL remnant, IDL. And besides the LDL lipoproteins, what are the other lipoprotein that has a ApoB100? So thinking about, the ApoB100, where is the ApoB100 located? In which type of lipoproteins? Then you can thinking about, which type of lipoprotein levels would be affected, right? So ApoB100 located in LDL, right? And also IDL, right? VLDL. So deficiency in ApoB100 not only affected LDL but also VLDL, remnant IDL, right? So what about VLDL remnant levels in this patient? It should also be high, right? So again, ApoB100 deficient affect both LDL and VLDL remnant IDL, and the LDL and IDL cannot be efficiently picked up by the cells, so remain in the circulation, so causing increased levels of LDL and VLDL remnant IDL. And plasma LDL cholesterol levels in patient high, right? Yes, high, because you have a lot of LDL remaining in the circulation. LDL basically is transporters of cholesterol, right? So cholesterol, LDL cholesterol levels high in the plasma, and risk for atherosclerosis. It goes up, right? Increase. So, and what is the mechanism? Why ApoB, for example, ApoB100 or LDL receptor deficient patient have increased atherosclerosis? Thinking about the three main reasons for the development of atherosclerosis. High LDL levels, right? Because they have a high LDL levels, and high LDL level is one of the main three reasons for the development of atherosclerosis. And you're probably wondering, because LDL is picked up by LDL receptor, right? If you have an LDL receptor deficiency, you should not be able to pick up LDL into the cells. Why LDL? I mean the cholesterol levels high in the accumulate high in the macrophages. Remember? Remember I mentioned that the mechanism for the development of atherosclerosis, the LDL, increased LDL is oxidized first, right? And oxidized LDL picked up by scavenger receptor, which is independent of the normal LDL receptor pathways, right? So a high amount of LDL can be picked up by the scavenger receptor and the macrophage cells, right? Okay, so the last case is the other type of hyperlipoproteinemia that caused by the defects in the ApoE. So ApoE, we know also, is the other type of apoprotein, right? It's required for the recognition of the LDL receptors, right? So what happens to the patient in terms of deficiency in remnant clearance by the liver, and chylomicron and VLDL remain in the circulation. So also, first of all, you need thinking about which type of lipoproteins have ApoE, and so ApoE deficiency should affect this type of ApoE containing lipoproteins, right? So basically, VLDL and chylomicron are the two type of lipoprotein that have a ApoE on their surface, right? So ApoE deficiency should affect both chylomicron and VLDL. So without ApoE on chylomicron or VLDL, and chylomicron VLDL cannot be picked up, right? Remember, by the liver, particularly chylomicron remnant by liver, or the VLDL remnant by the liver. So the patient with the ApoE deficient should have a higher levels of chylomicron and VLDL remnants in the circulation, right? Does that make sense? So, and what about IDL and LDL levels? It's also increased, right? Because this IDL particularly also have ApoE, so affected, and plasma cholesterol levels increase, and atherosclerosis development. The risk is also high, right? And the reason it should be the similar that I just mentioned because of high levels of LDL, right? All right, any questions? All right, so I think we finished all the lipid metabolism except for essential fatty acids. I'm going to talk about this Thursday. And let's see what else we have. Not sure. Do you guys have this slide? Oh, okay. Yeah. So this you probably better, sort of summarize the what type of lipoprotein has a different, ApoE, B100, ApoC2. So that will help you to understand if deficient of ApoE, ApoB100, or ApoC2, and which type of lipoprotein were affected, or which type of lipids, triglyceride, or cholesterol were affected, right? Okay, so I guess it's quiz time, right?